Hello everyone, hello. Today we'll talk about protein folding. Let me see who all are online, and we're going to talk about protein folding, which is an important topic in IIT JAM. Okay guys? तो कौन कौन ऑनलाइन है बता दो, थोड़ा हाई हेलो कुछ तो बोल दो ताकि मेरे को समझ में आ जाए कि आप लोग हो मेरे साथ. Let me see who all are online. Okay guys? Okay, we'll wait a little bit and then we'll start. So today we'll talk about protein folding, okay? And this is a small topic, important topic. 1-2 questions are always asked. तो इसके बारे में हम लोग अच्छे से पढ़ लेते हैं. ये बहुत ही important topic है from a concept point of view, and you need to know this concept regardless whether it is asked in exam or it is not asked in an exam, doesn't matter. Okay? So let us talk about protein folding. So when we talk about protein folding, what comes in your mind, right? So we should start with protein structure. We know that, okay, good afternoon Lakshmi, good afternoon. So when we talk about protein folding, what comes in our mind? The protein structure, right? And protein structure has different levels. First of all, it has primary level, secondary structure, quaternary structure, tertiary structure, etc. right? So primary structure है, इसके बाद secondary structure है थोड़ा higher order, then tertiary or quaternary structure भी है. So you start with a beads of amino acid, it's like a necklace of amino acid, and you achieve complicated structures like this. The whole point of protein folding is like how these kind of complicated structures are achieved inside a cell. That is all about protein folding. Anyway, let me tell you that my crash courses on IIT JAM 2021 is already present in Unacademy, and it is happening this month. So hurry up. If you haven't yet joined, do join or consider of joining, okay? It would be really interesting for you guys, and each and everything would be covered in these time span. Okay, so let me tell you what is this. So this is nothing, this is just a primary structure of protein, right? Primary structure of protein, which is nothing but a chain of amino acids. But what happens is this primary structure eventually changes, and they change in a multitude of manner, and what they really end up being is a 3D folded structure, right, which is functionally active. So if you don't have a native structure of a protein, your protein would not be functional. So protein structure is important for function, right? So let me write that protein structure is directly correlated with protein function. And our goal is to discuss about this protein folding part. The protein folding is not an easy process, right? But people started looking at it in early 1960s. And I'll tell you about Anfinsen's experiment. This man, Anfinsen, did a very simple experiment but a groundbreaking one. So what Anfinsen asked? Anfinsen asked, what is really guiding this protein folding process? So I mean, folding into this complicated structure, how it is possible? What are the factors that are governing these kind of folding kind of processes? Okay, is it crafted by sequence of events, or it is a random stochastic process that you end up being in a folded state, or you could go to multiple state and you can end up being in a folded state, right? So this was Anfinsen's question. So what he did, he took a native protein which was RNase A. So Anfinsen's experiment is directly in your syllabus and it is important. So Anfinsen's experiment says that if you put urea and beta mercap to ethanol, so beta mercap to ethanol get rid of all of these disulfide linkages. Now what happens? The protein gets unfolded like this. It is back to its primary structure, right? Now upon removal of this urea and beta mercap to ethanol, it gets back to its native conformation. So what happens really in this experiment is you have a native state, and this native state is catalytically active. This is very important. This native state, this 3D conformation is catalytically active. Now upon adding urea and beta mercap to ethanol, you see unfolding happened because all the disulfide bonds were broken right now. It is reduced because beta mercap to ethanol is a reducing agent, and that is why its 3D structure is now gone and it's just like a unfolded protein. Now what you can do, you can remove the urea and beta mercap to ethanol, which are the perturbing agent. And in this experiment, Anfinsen's found that native proteins are now, the native structure could be reobtained. Okay, the native structure could be reobtained. Right? So you understood this part? Yeah. Now let me tell you two things. So energy that is corresponding to protein folding can be subdivided into two kind of schematic. One is called the smooth landscape, one is called the rugged landscape. So this is a fairly ideal situation. It turns out that okay, when protein get back to its native structure, it's like a smooth process. Like it starts with a random structure, and there are only one or few ways by which it can roll down and get back to this native structure. So here is the native structure. Okay, here is the native structure. But the modern view says this is far more complicated than the simpler model. It says the protein folding landscape is basically a rugged landscape. So there are many kinetic traps. So imagine a ball which is rolling down this path, and if it is rolling down this path, this would be stuck here. See my pointer? It would be stuck here. That means that okay, there are kinetic traps, there are energy traps. Okay, so these are unfavorable energy states. Unfavorable energy barriers, I would say, than state. So this unfavorable energy barrier would prevent it to reach the native state. So it's like that game. So your protein, so each dot here represent a protein folded state. So it is unfolded, here the protein is unfolded. Now it would be folded here. Okay, so in order to do that, it has to really get to this native state point. But you know, this path is not really linear for these proteins. It's very difficult for them. इतना difficult है उसको, इतना difficulty को overcome करके प्रोटीन को fold होना पड़ता है, right? तो आपको लगेगा कि भाई आप ग्राफ में दिखा रहे हो कि ऐसे states है, ऐसे energy barriers है, energy traps है, जहां पे प्रोटीन जाके misfolded होके बन सकता है. मतलब क्यों? सेल के अंदर क्यों ऐसे होता है? क्योंकि देखो, अपना सेल के अंदर बहुत सारा मॉलिक्यूल्स है और वो मोशन मूवमेंट पे है, राइट? एंड दे आर कोलाइडिंग विद ईच अदर. मतलब प्रोटीन मानो थोड़ा सा फोल्ड हुआ एंड देन कोलाइड हो गया, देन फिर से थोड़ा सा अनस्टेबल हो गया और वो ठीक ठाक अपना स्टेट पे पहुंच नहीं पाया. तो हमेशा ऐसा प्रोसेसेस हो रहा है, राइट? दैट इज व्हाई देयर आर मेनी प्रोसेसेस जहां पे क्या हो रहा है, प्रोटीन स्टार्ट तो कर रहा है ठीक से, कुछ देर नीचे आने के बाद दे आर maybe they are going down, or maybe they are trapped, maybe they are trapped in these energy barriers, right? So what these energy profiles tells you is ऐसा कोई मैकेनिज्म है क्या जो इस प्रोटीन को ऐसे बैरियर को ओवरकम करने के लिए हेल्प करे? जैसे एंजाइम हमारा बैरियर को ओवरकम करने के लिए हेल्प करते हैं ना, तो एनर्जी बैरियर को, एक्टिवेशन एनर्जी बैरियर को एंजाइम लोअर कर देता है. तो प्रोटीन फोल्डिंग करने के लिए कोई एंजाइम लाइक कुछ सब्सटेंसेस है क्या? ये है एक इंपॉर्टेंट क्वेश्चन, राइट, जो आपका दिमाग में आना चाहिए. एंड हियर इज द स्टोरी ऑफ chaperones. chaperones are proteins which help in this process. So chaperones help to overcome this energy barrier. So let me tell you how. Look at this process in a dynamic simulation. So this particular black dot here, it is representing a protein. So when it is folding, and in the left side there is a simulation how protein is folding. When it is folding now, it can get to many energy states, and the lowest possible energy state is your native structure, which is the most stable state. But getting to that is a stochastic process. But chaperone makes it way more easier for these proteins to achieve a native state. So chaperone is like a catalytic enzyme in this process. It's not exactly like an enzyme, but it has catalytic power. Okay, so chaperone is helping in this process, augmenting this process of protein folding. And it makes sense because inside the cell there are so many processes that are happening at once, and so many molecular collisions which is preventing these kind of protein folding, right? And that is why you need some guides which are chaperone. So what chaperone really does? Chaperone, first of all, help to overcome this. Chaperone helps to overcome energy barrier. Okay, chaperone also help in refolding. Let's say one protein, due to some mistake or due to some chance, a protein is misfolded. What chaperone would do inside the cell, it would try to refold that protein back to the native conformation. Now if you know that there are many irreversible states, when you reach there, even chaperone या फिर chaperone का associated proteins or helpers can do nothing, and that is amyloid fibrils or oligomers, amorphous aggregates. So once aggregate is formed, chaperone really cannot do anything. So but chaperone can really change the partially folded state to a folded to a properly folded state, or chaperone prevents for aggregation. For example, in Alzheimer's disease, the biggest problem is aggregation of proteins. Chaperones help in aggregation. Okay, so sorry, chaperone prevents this aggregation process. This is a simpler version what chaperone does. So after protein synthesis, protein need to be folded properly into its native state. So this is its native state, and in this native state the protein is functional. If the protein is misfolded, unfolded or misfolded, it would go to an aggregate formation which is non-functional. Okay, now this aggregate can not only increase the inflammation, but it can also do many adverse effects. We'll tell you what happens to misfolded proteins. At the end of this lecture, there would be a consequence of misfolding. I'll come to that. Now what happens is, the unfolded protein shows folding, but it can also go to different routes and lead to aggregation. But chaperones prevent this route. Chaperones actually help this unfolded protein or guide this unfolded protein to a folded state, right? So chaperones guides unfolded protein such that it can fold properly, and chaperones prevent. So aggregation process, aggregation is inhibited by chaperones. Okay, aggregation is inhibited by chaperones. Okay, now what happens is, so this is the overall framework. If you understand this part, you understand the protein folding 50%. Now let me tell you our classification of chaperones. Now chaperones are widely classified into many subdivisions. Okay, so the chaperones So imagine this is an unfolded protein and they would have many hydrophilic, hydrophobic amino acid. Now this hydrophobic amino acid should be inside in a native conformation. Now what chaperone does, chaperones are two types. One is molecular chaperone or chaperoning. These are different in structure. Each does different functions. Chaperones work with the help of ATP hydrolysis, okay? So chaperone does what? After the protein which is synthesized from the ribosome, this chaperone quickly binds to these hydrophobic parts or other parts of the protein and they guide this folding. Instead of this folding to happen in a stochastic fashion, which is already a way, they guide this folding. For example, you don't know a route and you are walking down the route. It's first of all very slow process, right? And if somebody guides you, it would be easier. So chaperonines are other type of chaperone. So they are HSP60 family chaperones. Now there are proteins which can get inside this chaperone. This is like a vessel. Inside this, ATP hydrolysis lead to proper folding. So this chaperone change in conformation and just spits out the properly folded protein. You understood there are two different modes by which chaperone can actually fold protein. Now there is no right or wrong that which mode is right or which mode is wrong. Both of these modes are found in animal cells, or this kind of mode is highly chaperoning mediated folding are highly found in bacterial cells. Okay, so it is like you are going to a particular path. You don't know the path, so you would look at Google Map and slowly go there. There are multiple bends and etc. that might mislead you your path, right? But if you have a Uber which knows, okay, this is the path and I would take you and guide you to your destination, then you would smoothly reach your destination. Exactly. Chaperone do the same. They take the protein from an unfolded state to a folded state very smoothly down the energy landscape. Okay, so such that the protein gets folded to its native state. So it's like a molecular Uber or Ola cab, right? So it is really taking it to the nice destination. Otherwise, it might have mistaken its destination. It might have been lost in other alternate conformations which are not active, right? So this is the importance of chaperone. At any point of time you want more details, more detailed course, you can go and visit my lectures in Unacademy, which is India's biggest learning platform, and using my code AP10 you can get a 10% discount, and the ultimate course for IIT JAM is now happening. So go and watch it. Okay, so now let me tell you, in Alzheimer's disease, which is a neurodegenerative disease, there is abrogation in chaperone mediated machineries. Like chaperone does not function very properly, and with other associated reasons there are aggregation of proteins, and these are called beta amyloid plaques. So these are unfolded aggregated proteins. So aggregated proteins already tell us that they could cause consequences and they could be detrimental in terms of neurodevelopment. Now let me tell you, forget about neurodevelopment or disease angle. It is happening all the time inside our cells. So we are right now in the endoplasmic reticulum. So we are looking at one particular endoplasmic reticulum cisternae, RER obviously, not ACR. So inside on the top of RER you have ribosomes, which is synthesizing proteins. Just after the synthesizing proteins, with the help of signal recognition particle, proteins move inside, right? But in an unfolded state. Now once the protein moves in an unfolded state, it gets recruited with chaperones such as calnexin, calreticulin, etc. So this chaperone help in proper folding of proteins inside the endoplasmic reticulum. Now let me tell you that why it is so important. Now once a protein is totally folded, then it is termed as a functional protein, right? Otherwise it would be a protein but not functional, and non-functional proteins are not useful for the cell. So ER works like a quality control. ER chaperones work like a quality control. It ensures until and unless the folding process is complete, the protein should not leave the ER. And how does it ensure it? There are specific tags. There is a glucose tag which is ensuring the chaperone should bind to this protein. Once this glucose tag is removed, Now the protein can easily move out of the ER and go to its destination. But let's say ER found that one particular protein is misfolded. Then and there it would put this tag again, such that the chaperone says, okay, no, something is wrong with this protein, it is not properly folded, I should try to refold it, and then I would send it. So again it would engage. So that was one molecular function of chaperone. Now we come to another molecular function of these chaperones. In case of steroid signaling, the steroid receptor is kind of sequestered by the chaperone HSP90 in the cytoplasm in an inactive state. Now when there is ligand binding, which is steroid hormone, these chaperone disengage. So sometimes chaperone just hold the protein in particular location. That is also the function. And now this particular receptor ligand complex can move to the nucleus where it can lead to gene activation. So in cell signaling, chaperones are also important. So don't think that chaperones are only some machineries which can hold proteins. When it is concerned with signaling, chaperones also play vital roles. Okay. Now let me tell you the most important part, prion disease. So we were bothered about protein folding, right? So why should we bothered about protein folding? 4-5 प्रोटीन मिसफोल्डेड हो गया, डिग्रेड हो गया, तो हमको फर्क पड़ता है क्या? हाँ, फर्क पड़ता है, सेल को फर्क पड़ता है. Because it turns out there are some protein which are called PRP found in neurons, and C stands for their cellular, and there are aberrant variation which is known as PRP-SC. SC stands for scrapie in sheep. I'll tell you what scrapie is. Now this PRP-SC version is a aggregated version. It is not a proper conformation, it is not functional and all of these things. So what PRP-SC does, PRP-SC sequester native proteins and also misfold them, also induce misfold them. So ये ऐसा situation है कि मैं तो खराब हूं, मैं तेरे को भी खराब कर दूंगा, ऐसे situation है, ठीक है? So that is why prions are very very scary. Okay, it is not really known that how this conformation happens. One of the speculation is chaperone mediated mechanisms are not really prevalent, and somehow there is an abrogation in this mechanism. That is why this misfolding happens. And there are many diseases such as Creutzfeldt-Jakob disease, there is a variant Creutzfeldt-Jakob disease, Kuru disease, etc., which are, or mad cow disease, these are all due to prions. Okay, yeah. So that really tells us the importance of prions in our disease angle, right? So prion makes the brain really spongiform. So what happens is it's called bovine spongiform encephalopathy. Okay, so in this state your brain becomes like sponge. So your brain is really degraded, in a degraded situation, in a very bad condition. Okay, so that is why you need to understand this part. Okay, that why protein folding is very very important. If protein folding is not important, if protein folding doesn't happen, then we would not have folded proteins, we would have problems. Okay guys, yeah, we would have problems. That is why protein folding is very important, and we learned about protein folding in this short lecture. The detailed lecture is present in Unacademy. You can join my course using my code AP10 to get 10% discount, and once you access that course you would get a detailed overview of all of these processes. And this is just a trailer guys, picture to abhi baki hai. Anyway, I hope this was helpful, and please share this lecture with your friends, they would get benefit out of it. This is totally free, and do subscribe Unacademy's YouTube channel, and all the description regarding my courses, my profile is given in the description. Take a look, and you can also subscribe using that link. Anyway, I'll see you guys next time. Until next time, bye bye.